here just kind of talking about, you know, this breakdown of molecules or building up of molecules, the enzymes, is one way in which we categorize organisms and kind of give them a profile by running all sorts of different tests, which is those biochemical tests, which we will be doing for the unknowns, okay? Now, so we just went through really the focus of the glucose molecule, specifically. It's going to enter into glycolysis and go on either through fermentation, stopping with glycolysis and dropping off those electrons with the end product, or for cellular respiration, we'll see the Krebs cycle followed by the electron transport chain, right? All sugars, all your carbohydrates are going to go in the same direction, right? A polysaccharide is just going to be cut up into smaller pieces, to simple sugars, so that we get that glucose. If it's a different simple sugar besides glucose, then what we'll have is a reorganization of that so that it can move in order to move in or enter in some stage of glycolysis. Okay, so there's several, like for instance, fructose is a stage. The fructose 1,6-bisphosphate is a stage within glycolysis. So if you're looking at fructose, and we're trying to metabolize that, one could have 2-phosphate added to it, and it enters in at that location. So carbohydrates are going to enter in usually somewhere within glycolysis. Now, what about our other macromolecules? So we're going to focus on talking about the lipids and the proteins, okay, as far as where they are going to step into catabolism. So when we look at lipid catabolism, we'll focus on probably the easiest of the lipids, because lipids is a pretty, they're all monomers really, right? So when you break it down, you're breaking into subunits of that monomer, and it's a pretty diverse group, right? But generally for storage, energy storage, it's in your fat molecules, right? So the triglycerides. So for a triglyceride, you're looking at a glycerol with 3 fatty acids attached, okay? So we have the enzyme lipase that is going to remove the fatty acids from the glycerol, and then glycerol will actually enter in that the glycerol into glycolysis. Excuse me. Glycerol will enter in through glycolysis as a 3-carbon molecule. Okay, so it is going to enter in right where the split, that 6-carbon splits into 2 3-carbons. Now the fatty acids are going to go through a process called beta oxidation. And beta oxidation is where the fatty acids can be very long, and they're just going to be chopped up by 2s. Every 2 of this carbon skeleton is going to be removed, and is going to then be attached to acetyl-CoA and enter into the Krebs cycle. Now, based upon our quiz, we all now remember that the Krebs cycle is known for the production of, not ATP, the electron carriers that will go to the electron transport chain, right? So when those 2 carbons are added to coenzyme A to give you acetyl-CoA, those are just going to, every 2 then are going to enter into the Krebs cycle, and we're going to get those electron carriers, FADH and, FADH2 and NADH, okay? And we're going to be cranking out a lot because fatty acids can be very lengthy. And there's 3 of them. And then all those electron carriers, they move to the electron transport chain, and we get ATP produced. Okay, so that kind of explains then why triglycerides are a big storage mechanism for energy, for a cell. All right. Then as far as protein catabolism, well, proteins, we have that polypeptide. So the first thing we need to do is break that polypeptide back into the amino acids. So we have various proteases that are going to be involved in that process. So the example in this particular picture shows the protease caseinase, which is going to break down casein, which is the protein for milk, the white protein found in milk, right? Because that white characteristic or color. Collagenase, gelatinase, or some other examples of proteases that are going to break down those specific proteins. So we have different proteases. We have some proteases that will cleave proteins in general, and some are more specified to a particular protein. Okay. So collagenase will break down collagen, gelatinase is going to break down gelatin. Now I bring up these specifically because these are some of the proteases that one would look for and with those biochemical testing when they're creating that profile for that microorganism. We used to, with our unknowns, we used to look at caseinase, so there would be a milk agar, and you would put your organism on there and then see if it broke down, if it had a clearing in the media around that colony of growth to indicate it was breaking down the white milk protein, casein. We don't do that anymore, but we do the gelatinase test later in this term for the unknown. So that'll be looking to see if our particular organisms produce gelatinase, which breaks down gelatin, right? So those are just different items that we look for. These proteases are going to break down to that protein, that polypeptide, into the amino acids. Once we have the amino acids, then we have the first thing that's going to happen is a process called deamination. It's going to deamination removes the amino group. So the amino group is going to be removed, and then what's left behind, that is going to go through various stages, more often than not in the Krebs cycle. It's going to enter somewhere in the Krebs cycle. So this is just a picture. I don't know if this one came from your book or whether I pulled it off. I think I pulled this one offline. That actually, and you would not have to know, but it does actually list where the different, the amino acids enter in once that amino group is removed. So some enter in within the range of pyruvate as a 3-carbon molecule. Some are actually going to enter in as that 2-carbon with acetyl-CoA, right? Some actually enter in, this is the oxaloacetate is the start molecule and end molecule of the Krebs cycle. So some are actually going to move in and enter the system through that route. Others through other, molecules in that Krebs cycle that occur. Okay, so again, it just depends upon the particular protein as to where it might enter into that process. All right. That was the last of the slides, okay? For the module 5, so let's go into module 6. So yesterday I did, I remember I said that test your knowledge. I found a little sheet on the cellular respiration. So I did actually put that in, and I changed the title to say that's specific in the test your knowledge and for the intro for module 5, for module 5, I did actually add, it's a little diagram type of chart. So it's more of a visual learner type of setup as far as cellular respiration is concerned, okay? Did anybody happen to see it yet? No, so a couple of people did, okay. Yeah. There's a word bank, and you kind of place the words within that visual diagram of what's happening where. Okay. All right. So for module 6, we're going to go into microbial growth and nutrition. So for microbial growth, unlike when we talk about say a multicellular organism, when we're talking about a single-celled organism, microbial growth really refers to population increase in size, right? not the size of the organism growing, for instance, infant to adult, right? Because, yeah, with multicellular organisms, you do see that type of change, usually is a size during the lifespan of the organism. But for single-celled organisms, it's not necessarily the size. Now they do, increase in size during that process of, as they're preparing for cell division. But other than that, during their lifespan, they're the same size. Okay, so yeah, growth is that increase in population. And of course, the increase in population is due to reproduction. Okay. When we look at microbial growth, you've already seen it in the laboratory. So when we look at microbial growth, if you're looking at a broth, you're going to see it as the change in the optical density. It's going to become higher optical density, more turbid, cloudiness in that media. When we are talking about microbial growth on agar surfaces, we're generally talking about a distinct, discrete colony or aggregate, so this group of cells or aggregate of cells that arise from a single cell or a group of attached cells, okay, that produce that colony. We also have something unique too when we talk about microbial growth, is a unique population referred to as the biofilm. Now, the biofilm is a collection of microorganisms living on the surface, and that biofilm is one in which is a mixed culture of organisms that are working together and living and sustaining that community. When microorganisms are part of a biofilm, they lose some of their characteristics that they might have when they are free living, okay, because they become part of that community. A biofilm can consist of prokaryotic cells and eukaryotic cells. Of course, the easiest description of biofilm would be plaque on the teeth, okay? Or if you look at your shower curtain or around your plumbing, right? That slime layer that might start to, build between your, day cleaning and the next day cleaning. And almost every pipe, plumbing pipe work has inside of it a biofilm that develops, okay? That is a mixed culture of organisms that are sustaining life. All right. So, all right, so for this particular module, our goal is, first of all, to classify organisms based on carbon and energy source. You did this in general biology, so we're going to repeat that process, right? Of remember that, your autotrophs and your heterotrophs, etc. Right? So hopefully that won't be anything new when you go through that particular material. And then we're going to actually then start looking at differences between organisms based upon their oxygen use and tolerance. We all think that cells are like our cells, and that all cells need oxygen, but that is not the case. Okay, and there are microorganisms that actually die in the presence of oxygen. They cannot live in the presence of oxygen. Many of the organisms that live in your gut are these anaerobic organisms, because the gut in your gut is low oxygen presence, so it's pretty close to an anaerobic environment. Okay. All right. And then we're going to classify microorganisms based on various environmental conditions or requirements that they might have in order to sustain growth. And then we're going to go through actually the formation of that biofilm and how it works. We're going to talk about binary fission, so the process by which bacterial cells, so your prokaryotic cells, divide. Right, we'll talk about generation time, and then we're going to go into the phases of microbial growth. We will also, you'll notice in this unit, they'll talk about enumerating microorganisms. So since we did it in lab, we'll touch upon different ways we can enumerate organisms here too in the on the lecture side. Okay, so you'll see that in this particular module. All right, so organisms are of course going to use a variety of nutrients for their energy needs, right? They're going to require this to build those organic molecules that they need. Those necessary elements, there are 4 required elements for organic molecules, where 96% of the cell is composed of these 4 elements. Do you know what they are? Carbon is an obvious one, good. Oxygen, nitrogen, and hydrogen. There you go. I'm sure somebody said it, I just didn't quite hear it. So those are your 4, okay? Sulfur and phosphorus is are also 2 that are going to be important also in that group. But the top 4 are that carbon, oxygen, nitrogen, hydrogen. Okay. Now, microorganisms are going to obtain these particular elements from different sources, and so here we're going to be looking at the sources where they contain are going to get their carbon. Okay. We'll also talk in the process. We'll also be incorporating where they're getting their energy from. Okay. So we have when we're talking about energy, the energy can come from either chemicals or from light. Okay, light then of course would be where they're taking it's through photosynthesis, right? Light, if they're obtaining their energy through light, that would be your phototrophs, which is shown on the bottom half. So we're over here on the bottom half. And then if it's getting energy from chemicals, so they're they metabolize chemicals to obtain their energy, they're chemotrophs. Okay, the phototrophs are known, so they're pulling the light from they're pulling light to produce their energy, excuse me. So now we are going to look at where they could be getting their carbon source from. If their carbon source is from inorganic carbon, they are referred to as autotrophs. And so we combine those words to say photoautotrophs. Okay, so oftentimes we're looking at both the combination. We tend to be looking at this terminology that tells us both energy and carbon at the same time. Okay, photoautotrophs would be light for your energy source and inorganic material for your carbon source. If we are looking at autotrophs that are getting their energy from chemicals, then it is a chemoautotrophs. Autotrophs are known as your producers, okay, because they can take something inorganic and bring it into the organic world. Any other organism that can't do that is referred to as a heterotroph, okay, the consumers. They have to get their organic material, their organic carbon, from a organic source, which means another organism, okay, whether that be, we're not talking about carnivores versus I'm a vegetarian or that type of thing, right? Because fact of the matter is both plants and animals are living organisms, and they're both organic organisms, and therefore no matter which one you consume is still organic material you're consuming. Okay, so now when we combine those, we have the chemoheterotrophs coming, so chemical is providing the energy, organic material is providing the carbon, or we have our photoheterotrophs. Now in general biology, this one, chemoheterotrophs, right? And photoheterotrophs were probably your biggest focus when you're talking about these different groups. And that is because all animals, right, fall into this group and all plants into the other group. So when we're looking at our eukaryotic cells, you'll notice eukaryotic cells are really falling into one of the two, photoautotrophs as a producer or chemoheterotrophs as a consumer. Okay, the other two, your chemoautotrophs and your photoheterotrophs, these are going to be coming from your microorganisms. So most of your prokaryotic cells have much more diversity as far as how they can handle their environment and live in their environment. Okay, than these much more complex but limited eukaryotic cells. Okay, all right. Now, we can also look at, and now I'm not about the electrons. We'll skip over the electrons. The book doesn't cover it so much, so the text we're following, so I'll let that one go. As far as, so now we move into our oxygen, right? Our oxygen requirements. Oxygen is the next next one on our list of critical elements that cells need for their macromolecules. Oxygen, though, has a little bit of a problem that coexists with that particular element, and that's the fact that it's highly electronegative. Okay, and so we have when cells are metabolizing in the presence of oxygen, there are toxic forms of oxygen that are going to be produced. The toxic forms of oxygen are singlet oxygen, superoxide radicals, peroxide ions, and hydroxyl radicals. Those toxic substances, what they are, they're oxidizing agents. Okay, remember those redox reactions. LEO lose electrons is oxidized. They're going to snatch electrons from other molecules. Okay, and this oftentimes results in irreparable damage to the cells. So, how do the cells combat against that? Obviously, we live in the presence of oxygen. Do our body and our cells have to deal with this? Absolutely. Okay, any cell that's metabolized in the presence of oxygen is going to have to deal with this. So, how cells deal with this is to produce enzymes that detoxify it. Okay, so there's an enzyme called superoxide dismutase that breaks down the superoxide free radicals into hydrogen peroxide and oxygen. Unfortunately, that's not going to be sufficient because hydrogen peroxide is one of the toxic byproducts. So that's not going to be an endpoint. Just having that's not going to be enough. We have two other enzymes that can break hydrogen peroxide. We have catalase and peroxidase. Catalase, when it breaks down hydrogen peroxide, produces water and oxygen as an end product. And peroxidase, when it breaks down hydrogen peroxide, produces just water. Okay? So a cell who's going to, or an organism who's going to live in the presence of oxygen is going to need to be able to detoxify those toxic products. So they're going to need to have several of these enzymes. At minimum, superoxide dismutase and one of these other two. Right? Otherwise, the presence of these toxic byproducts are going to become a problem as far as that cell is concerned and living in the presence of oxygen. Now, there is actually a biochemical test that we perform, and we will do it later this term in the lab, which is the catalase test. And for the catalase test, all we're going to do is put our organism directly on the slide, no water, just organism, smear on the slide, okay? And then we're going to get hydrogen peroxide, 3% hydrogen peroxide. We're going to put drops of 3% hydrogen peroxide on it. If catalase is present, remember catalase breaks down hydrogen peroxide into water and oxygen. Oxygen is a gas, okay? So oxygen is going to rise and create those bubbles. This indicates that the organism produces catalase. If it does not produce bubbles, that indicates that they do not produce that enzyme, so we refer to them as catalase negative. Now, most of you sometime in your life, maybe not, but I'm guessing that most of you at some point in time in your life have had an injury, a cut, nick on the skin where you might have put hydrogen peroxide on it, right? So this is, why? You put it there for a reason. The reason was to, as an antiseptic, right? To remove any potential pathogens that may have entered in through that nick or cut when it occurred. You've got a scratch that broke the skin. We want to make sure that we remove any organism from that location. And of course, it started bubbling, right? If you've done it, you've seen it. If you haven't done it, don't go home and make yourself a cut and do it, okay? But if you get a cut at some point, try it. It's the better alternative than using, as far as not stinging, than using alcohols, right, on a cut, because alcohol it stings. This doesn't, okay? But what's going to happen is that you're going to see bubbling, and that's not because of microorganisms. That's because your cells produce catalase. Okay, so your cells that were damaged, the enzyme is in the environment, and that hydrogen peroxide is being broken down by the enzymes you produce of catalase. After a time, of course, you generally, if you've done it before, after a time you notice what? After the bubbling stops, what happens? What's left? It's like a white film, usually. Why? Well, because you've killed off some of your cells in the process, right? But your cells will be able to be replaced. A bacterial cell, a single-celled organism that died off in that process too, is not going to be able to replace itself. Once it's dead, it can no longer produce more. Okay? Now the downside is, as one term I had somebody who worked for a dermatologist, and they said the downside though is because you are killing off some of your cells, the dermatologist wouldn't necessarily recommend this. They go for the alcohol because of the fact that you can get more scar tissue because you're damaging more of your cells, right? A nice clean cut, isn't going to get as much scar tissue production as you would if you had. It goes when you use that hydrogen peroxide. Now you killed off more cells, and it's not as nice and clean cut. Okay. All right. So, oxygen requirements. How do we determine oxygen requirements for these microorganisms? It is a biochemical test that we are going to be performing as far as what are the oxygen requirements for a particular microorganism. And there's a specialized media. We will be doing this in the next, I think we're starting it today, actually. Within the next week, okay? And the media is fluid thioglycolate medium, okay? So in lab, today we're going to be working with this, and of course abbreviated would be FTM. Okay, so fluid thioglycolate medium is a special type of media that has glucose present in it. It has sodium thioglycolate also, and that sodium thioglycolate is going to reduce oxygen, making it inaccessible. Okay, as a result, right, you create an environment where the top of the tube, closest to the atmosphere, the oxygen that's present in the air, is going to be higher and is going to have oxygen present because oxygen is always going to diffuse in. But at the base of the tube, it'll be anaerobic. Okay, because this media, the chemical in this media is going to be reducing that oxygen, making it no longer O2, right? So we're getting that reduction is going to take away its power of serving as that molecular oxygen, but you can't stop diffusion from happening. So diffusion from the environment is always going to continue. That's why the top of the tube is going to have some oxygen, but the bottom of the tube will be anaerobic. And then what you're going to do is add your specimen, right? Let it incubate and see where it grows in that tube. If it is growing solely at the top, as you see here, that is an obligate aerobe. These are organisms that are involved in cellular respiration, and they require oxygen for metabolism, and they're going to have those enzymes necessary to break down the toxic byproducts. If you find that the organism is solely growing at the bottom, which I hate when they do these because it's not representative truly of what you're going to see. Usually the organism grows all the way here. So the organism is turbid, all this area. It's clear at the top. This is the region where the oxygen is present. Okay, so growth is throughout, but not at the top. That is an obligate anaerobe. These organisms cannot live in the presence of oxygen. They are going to perform fermentation, okay, or anaerobic cellular respiration, one of those two. And they don't have those enzymes necessary to break down the toxic byproducts. We have the facultative anaerobes. The facultative anaerobes are going to grow throughout. They're going to tend to grow better at the top, but they'll be throughout the entire tube. These organisms are the ones that can alternate between, for metabolism, they can alternate between cellular respiration and fermentation. Okay, so they can pop back and forth based upon what's available. If oxygen is available, it's cellular respiration. If oxygen is not available, they're doing fermentation. They do have those enzymes necessary to live in the presence of oxygen, those toxic byproducts of oxygen. Okay, so they are going to grow throughout the tube. We also have what we call aerotolerant anaerobes. The aerotolerant anaerobes are a group of organisms that do not use oxygen for metabolism. Okay, so that is anaerobic cellular respiration or fermentation, no oxygen at all. But they still have the enzymes, so they can tolerate the presence of oxygen. They just don't use it. Okay? And then we have our microaerophiles. Our microaerophiles are going to grow in a band, not at the top, but not at the bottom, because they do require oxygen for metabolism. But they can't tolerate the levels of oxygen that we see atmospherically, which is, about 20%. They need to be much lower. Okay, maybe 10%, 2%. Okay, but wherever this tube, in this tube where we have that level of oxygen is where you're going to see them growing. Because they use oxygen for metabolism, they can't grow down here because they need it for their metabolism. They perform cellular respiration. They just can't tolerate higher levels of oxygen. Okay, so those are your microaerophiles. All right. Now, using fluid thioglycolate medium is great in the laboratory to determine oxygen requirements, but when you're trying to grow microorganisms on a petri dish, this isn't, or on a solid surface in order to isolate different organisms that exist out there in that particular environment, it's not going to be helpful, right? Because it is a liquid matrix. So for the purposes of growing in an anaerobic environment on a solid surface, we have our anaerobic, I call it an anaerobic chamber. I can't remember what they called it exactly in the text, but this is what I'd consider an anaerobic chamber. This one actually has got the ability to monitor it with a gauge, okay? And it almost looks like they've got to set it up for even transfer of oxygen using maybe even a tank. But in a anaerobic chamber, you put your specimen in. Many of them actually have these little packets, and we will see these are either this one today or the next lab, so next Monday. But they have little gas packets, or that have chemicals in them that will reduce oxygen. So you open it up, you add a little bit of water, there's a gas pack, and when you seal it, it's going to remove the oxygen, making it an anaerobic environment. So that while this is in the incubator, they sit in an anaerobic environment. You can also have an incubator that is anaerobic, right? Or if you're working in the laboratory setting, you can have, this is also a anaerobic chamber, right? But and so this environment is without oxygen.
They call one a jar and the other one a chamber.
Yeah, see, but I always called this the chamber myself, so you can call it the jar, chamber, whatever else. I don't care. A chamber is a containment area. So now, back when for anaerobic environments, this text brings up actually when they're talking about higher levels of CO2, but they also use it for anaerobic environments, is the candle jar. So they would have, because they hadn't designed yet the technology for the chemicals and blah blah to remove the oxygen. So what they would do is take a big glass jar. They would put their petri dishes in the glass jar. They put a candle in there, light the candle, close the lid. Right? Candle's going to burn until oxygen burns out. So once oxygen is no longer in that candle jar, it's become an anaerobic environment. It also, though, is higher in levels of CO2. So that's where they in this text they're bringing up the candle jar. Okay, is the fact that when you have organisms that want low oxygen but high levels of carbon dioxide, then that is an option. Okay. Many organisms are going to prefer, intermediate oxygen levels. And some do like high CO2 levels. In fact, the body tissue, any area of the body tissue, it tends to be higher in CO2 than oxygen. So the organisms that grow and cause disease in body tissue are going to be ones that enjoy that and grow well in that type of environment. If they grow really well in high carbon dioxide levels, they kind of almost require high levels of CO2 as compared to oxygen. Those are referred to as our campnophiles. Okay, so campnophiles, need that high level of oxygen. Of carbon dioxide, over oxygen. All right. Other conditions that come into play for the microorganisms and their growth requirements. Well, the pH of an environment. Most of our microorganisms are going to fall into a group referred to as the neutrophiles. Okay, the neutrophiles are going to be in a range, around 7. 1.5 pH levels from that, basically. Okay. Some organisms are going to prefer a little more of an alkaline environment. So if they require an alkaline environment, they're called the alkalinophiles. If they require an acidic environment, they're called the acidophiles. Usually when you go towards the acidophiles and alkalophiles, we're looking at those more extreme environments. Okay, and therefore, yes, some bacteria, but also your archaea. They're gonna oftentimes fall into these groups. Acidophiles, they have recognized organisms that even grow, they are able to grow even at a pH of 0. So very acidic, the most acidic you can get. Okay. Now, when you look at these, you'll notice, just like we talked about with the enzymes, you'll notice that you have an optimum, right, pH for that organism, and then it splays out as a bell curve on either side. Okay, because a lot of this will end up having to do with denaturation of proteins. So these organisms that can tolerate the more acidic pHs or more basic pHs are going to have their protein structure is going to be organized a little bit differently in order to, maybe more disulfide bridges to hold that 3D level structure in the tertiary level, right? If you're talking about their DNAs or their nucleic acids, maybe they have more GC in them as compared to AT base pairing, because GC base pairing has 3 hydrogen bonds. The AT only has 2. So you're looking at more hydrogen bonding, which is just more strength in a way for those particular interactions between the double-stranded DNA. Obviously, for the most part, right, we're not really going to be worried about acidophiles and alkanophiles being pathogens for the human body. Okay, at least not for the bulk of, right, because we don't have a lot of environments. Most of the body environments are in this range here, between 6 and 8. Right, the stomach, yeah, okay, but the organism doesn't necessarily have to be an acidophile to be able to tolerate that level, right, of pH. It could be a neutrophile that just grows slower in that particular environment. All right, temperature. Temperature, we have the 3 cardinal temperature terms, 3 cardinal temperatures. That is the optimum temperature, the minimum temperature, and the maximum temperature. Okay, falls under the same general thought process as when we talked about the temperature effect on proteins. You'll notice these are less of a bell curve. Okay, the optimum tends to sit closer to the maximum temperature than it does to the minimum temperature for those cardinal temperature ranges. And we do have groups organized. We organized these microorganisms into groups based upon what their optimum range is. So the psychrophiles prefer a cooler temperature range, so their optimum temperature is going to sit between, say, 10 and 15 degrees Celsius, right? They are going to be able to tolerate in temperatures of, at or even below 0, and yet still have growth. As compared to then on the high end, once they move much higher than room temperature, you've already reached the max. Okay, so about max temperature is where there is about room temperature. The mesophiles are going to have an optimum temperature in the range of 30 to, in between 30 to 40 area. Okay, much higher than 40, we're already reaching our max temperature range. And they can grow in temperatures as low as, 10, 15. Okay, just not as well. right? Not as, they're gonna be growing much slower. The thermophiles have a maximum temperature range anywhere between 60, 70, sometimes it's close to 80. And then about anything after 80, they're usually tailoring off pretty fast for the maximum. They don't usually grow anything lower than about 40. And these are just general ranges here. And then your hyperthermophiles are going to grow optimally at somewhere above 80 degrees. Okay, that's their optimum is going to be above 80 degrees, and so they're going to be able to live in those extreme environments. So thermophiles and hyperthermophiles, you're looking at your hot springs, right? The bottom of the ocean near the thermal vents where the temperatures are much hotter. So those are generally, those prokaryotic cells that would fall in more of an archaea basis, okay? And then the psychrophiles, these are the ones that you're concerned about in the refrigerator, right? You have a couple, you put some, your refrigerator temperature is, standard is usually about anywhere from 4 to 10 degrees, right? 10's a little on the high side. Usually you would try and stick around 6 is what you're hoping for a temperature in the refrigerator, right? But we all know that we put some leftovers in the refrigerator and come back to it like a few days later, maybe if you're unfortunate enough and haven't gotten to cleaning out your refrigerator a month later. Yeah, I know, it happens. It doesn't mean you have to eat the food, right? Because when you go and open it up, you're like, oh, that's got to go right away, because you do see growth, right? So we do know in the refrigerator microorganisms do grow. They would be those psychrophiles, right? That are going to be growing in that. There's, let me make sure I have that. There is a term actually called psychrotrophs. So it's psychro and then TROPHS. The psychrotrophs are kind of the in-between. They sit kind of in between here and here. So they don't grow below freezing, but they sit right there. And those are the organisms often, actually more often than not, you're talking about psychrotrophs when you see something coming out of the fridge and it's growing, because their optimum temperature is usually around 20 room temperature. So while you have your food sitting out, cooling down, which you shouldn't do, right? You should put it in the refrigerator right away. But if it's sitting out, cooling down, then this is where microorganisms in the air, dust, attached to dust particle, whatever, land on the food, and then, they're able to still grow even though it's in the refrigerator. Of course, question for you, easy enough, I think, but maybe not. Which organisms are we going to be concerned about when we talk about human diseases? Which group? Mesophiles, right? If we're talking about human diseases, our biggest ones that we're going to be concerned about is the group of the mesophiles, because their optimum temperature is right around the same as ours. Okay. Make sure I put everything there. And we can do that one more. Physical effects of water. So water, too, the presence, the lack of water, of course, is one. So in very dry environments, they have a group of organisms called the xerophiles. Xerophiles is just like xeriscaping. It's, if you've heard of that, it's anytime you use plants that don't require a lot of water, right? The same thing here. You have some environments that are very dry environments. So these particular microorganisms that are referred to as xerophiles can tolerate those low, water environments in order to, and yet still be able to reproduce and population increase in size. You also, the other effect you have, another effect you have on water, of course, is osmotic pressure. So when we talk about osmotic pressure, we're talking about the solutes in the water. High solutes in the water, low solutes in the water. In general, for bacteria. It's usually they're in an environment that is hypotonic, right? And that's not, or isotonic. A hypotonic environment is not going to be a problem for a bacterial cell because of the cell wall, right? Water is going to, yeah. What is going out? No. Hypertonic. I see that's why I always have to draw the doggone things out. Water is going to be going in to, and the wall is going, so it's hyper, water is going to go, no, hypo. I gotta draw it out. I'm a very visual person. Hypo, right? You have a cell. Hypo means that the water's going in. There we go. I am right, first time around. So water's going to go in, but the cell membrane is going to hold the shape of that cell, because the cell membrane can't expand past that cell wall, right? So the cell membrane is being maintained by the cell wall. Isotonic, of course, water's going in and out equally, okay? But more often than not, these bacterial cells are in a hypotonic environment. It is the concern for cells, of course, is when they are in a hypertonic environment and water is leaving and the cell is becoming plasmolyzed. Well, some organisms have the ability to address that, right? That is your halophiles. So we already kind of started talking about that with the lab, and we'll be seeing some results of organisms that can tolerate higher levels of salt in lab today. Halophiles, and we have the term halophiles and halotolerant. We're actually going to be looking at halotolerant. Halophiles actually require that higher level. They're salt loving. They actually require the higher levels of salt as compared to halotolerant that can tolerate higher levels of salt. They also can be referred to as facultative halophiles, okay? Throw facultative in front of one of the phile words, and it just means that it can go either way. It doesn't care. You know, it can work. It doesn't have to have it, but it doesn't have a problem with it, okay? These are the organisms, the halophiles and the halotolerant. These are the organisms that we're, concerned about when you are, you have a situation where you're brining, pickling, and so you're using higher levels of salt to maybe inhibit microbial growth, because they're not going to be inhibited. Our body uses that system. Again, with perspiration and when the water leaves, what's left behind is the salts on the surface of the skin, which inhibit microbial growth, unless they're a halotolerant organism. Okay. And then, of course, we have hydrostatic pressure. Hydrostatic pressure has to do with the weight of water. So when microorganisms are in water, just like when you're in water, water has a weight. The deeper you go, the heavier weight it has on the system, okay? Creating that barostatic pressure. The barophiles are the organisms that like that need that live at the bottom of the ocean, right? Miles underneath water, so the huge amount of weight. And so you can't just take those organisms up from the bottom of the ocean and bring them to the surface and expect them to grow. They're not going to, okay? Because the cell is already designed in such a manner as to be able to survive in that particular environment. So it's along the same lines if you ever do the scuba diving, right? I don't want to see what I'm swimming with, but all my other family members have. And of course you have that, the concern of coming up too fast and getting bends, right? Which is life-threatening. So that's, where we're looking at when we talk about hydrostatic pressure. It's the actual weight of the water and the effect the cells have in order to accommodate that. So you can't just take it away and expect them to survive. It doesn't work. All right, we're going to stop here for today. past 7:15, so we'll make it 7:35 in the lab. There is no, unless you want one.
No.
Great. I feel like I'm not providing you with what you requested. Not at all. It was, what? We don't have a lab. At all? Not at all. You know, we don't have a lab. It's only gonna be big.